Hello everyone. Last time we have explained about the types of lysosomes and the lysosomal enzymes. Today we are going to explain the functions of lysosome. So lysosome undergoes several functions or performs several functions. So first function, important function performed by the lysosome, that is intracellular digestion. The lysosomes contain enzymes and digest the material intracellularly, called as intracellular digestion. So the lysosomal enzymes act upon the cytoplasmic materials of the same cell and causing intracellular digestion of the endogenous material or the cytoplasmic contents or the materials of the same cell. So intracellular digestion can occur by the lysosomal enzymes, and this type of digestion of the cytoplasmic materials is called as intracellular digestion. Intracellular digestion with the help of lysosome or lysosomal enzymes is of two types, such as autophagy and heterophagy. So autophagy is the intracellular digestion in which enzymes of the cell digest the pieces of cytoplasmic material from the cell itself. That is digestion of the endogenous material, and mitochondria are constantly removed by this process. It means together with the lysosomal enzymes, some mitochondrial enzymes or the part of the mitochondrion can be picked up by the lysosome, or there is a formation of autophagic vacuole or the lysosome called as autophagic vacuole, which are formed by the fusion of the certain portion of the mitochondrion containing certain enzymes with the lysosome or the primary lysosome to form autophagic vacuole, and thus the intracellular digestion of the endogenous material usually taking place with the help of enzymes supplied by the mitochondria or the part of mitochondria, and thus number of mitochondria reducing in the cell. So mitochondria are constantly removed or used for the process of intracellular digestion because the autophagic vacuoles or the lysosomes are formed with the use of definite part of the mitochondria containing number of enzymes. So all these enzymes are thus used for the intracellular digestion is called as autophagy. So autophagy or the type of autophagy or the type of intracellular digestion which is occurring in the cell itself and digesting the endogenous material of the cell with the help of enzymes supplied by the mitochondrion or the autophagic vacuole or the lysosome. So this is called as autophagy. Thus there is a renovation. And a turnover of cellular components. So due to the intracellular digestion of the endogenous material through the process of autophagy, there is a continuous renovation or the renewal and turnover of the cellular components. Means the old components or the undesired substances of the cytoplasm can be digested, and these are rejuvenated or renewed in the cell. So there is a renovation and the intracellular there is a renovation and the turnover of the cellular components due to the intracellular digestion by autophagy. Then second type of intracellular digestion, that is heterophagy, means intake of exogenous material into the cell and its breakdown or digestion by the lysosomal enzymes. So heterophagy means whatever the exogenous material or the various materials which are uh, introduced or entering into the cell by means of process of endocytosis, that is either pinocytosis or phagocytosis. So when there is a entry of liquid contents into the cell by means of process of endocytosis called as pinocytosis, and whenever there is a entry of solid materials into the cell, then is called as phagocytosis. So while heterophagy or during the heterophagy, whatever the exogenous material or the substances either in the form of liquid or the solid contents which are entering into the cell by the process of endocytosis, that is pinocytosis and phagocytosis, and forming either pinosome or the phagosome. So these either this pinosome or the phagosome get fused with the primary lysosome containing certain lysosomal enzymes in it. So the fusion of pinosome or pinosome and phagosome, either pinosome or a phagosome fuses with the primary lysosome containing certain enzymes to form a secondary lysosome called as heterophagosome or heteropinosome. So whatever the exogenous material that are trapped, that are absorbed, that are fused with the primary lysosome, and the process of digestion of the exogenous materials, either the liquid or solid substances, into the heterophagosome or a secondary lysosome called as heterophagosome, or a secondary lysosome taking place called as the intracellular digestion called as a heterophagy. So heterophagy means intake of exogenous material into the cell and its breakdown or the digestion by the lysosomal enzymes. Endocytosis is a phenomenon in which bulk of exogenous material is taken inside the cell, and it includes two phenomena such as pinocytosis, that is incorporation or entry of liquid materials, and phagocytosis, that is intake of solid materials. So the product of pinocytosis and phagocytosis, that is pinosome or phagosome, will enclose either liquid content or the solid contents which are brought into the cell, or these are the exogenous material, liquid or solid exogenous materials uh, received in the cell or enters in the cell to form pinosome or the phagosome. So this pinosome or the phagosome then fuse with the primary lysosome to form a secondary lysosome or a heterophagosome called as heterophagosome, and the process of digestion of the exogenous materials which have been trapped or fused with the primary lysosome to form secondary lysosome, the process of digestion will take place in the heterogeneous or heterophagosome or a secondary lysosome uh, inside the lysosome. So the heterophagy means intake of exogenous material into the cell and its breakdown by the lysosomal enzymes, and in this case, or while heterophagy or the intracellular digestion, usually the liquid or solid contents of the exogenous material are absorbed or fused with the lysosome, and then the lysosomal enzymes will act upon these exogenous materials and digest them inside the secondary lysosome called as heterophagy. Then second function of the lysosome, that is extracellular digestion. So the lysosomal enzymes are also released outside the cell and they hydrolyze the extracellular materials. Lysosomal enzymes not only causing the intracellular digestion of the endogenous material or the exogenous material in the cytoplasm of the cell with the help of lysosomes, but it also serves for the extracellular digestion, in which the lysosomal enzymes are released outside the cell and they hydrolyze the extracellular material. Lysosomal enzymes are also involved in the bone erosion. The example of the extracellular digestion, that is lysosomal enzymes which are involved in the erosion of the bone, bone erosion, that is the example of the extracellular digestion. Such a process may occur in the rheumatoid arthritis, a human disease in which cartilage of the joints may be eroded by the lysosomes. So lysosomal enzymes, when released to the outside of the cells, may act upon the cartilage of the joints in the disease called as rheumatoid arthritis, and this is a human disease in which cartilage of the joints are eroded or dissolved or digested by the lysosomal enzymes called as extracellular digestion. Then third function, hormone secretion. The lysosomal proteases activate secretion of thyroid hormones in the thyroid follicles. The lysosomal proteases, or that is the protein digestive enzymes, the lysosomal proteases activate secretion of thyroid hormones in the follicles. Then fourth function, lysosomes are important in germ cells and fertilization. Lysosomes are important in germ cells and fertilization. The acrosome of the spermatozoon or the male gamete is nothing but a special type of lysosome which contains the protease and hyaluronidase enzymes. The acrosome, that is a head region, and there is a acrosomal cap situated above the nucleus and present at the tip of the head of the sperm. So the acrosome of the sperm, which is present at the tip or at the anterior end of the sperm, in the tip of the head, just above the nucleus, is a type of special lysosome containing two type of enzymes such as the proteases and hyaluronidase, and these enzymes are useful for the penetration of the sperm into the egg and also for the digestion of stored reserve materials. So the enzymes of the acrosome containing the enzymes of the acrosome of the sperm is a special type of lysosome, and it contains two type enzymes, that is a proteases and hyaluronidase, and these enzymes are useful in dissolving the egg cell or the egg membrane while penetration of the sperm into the egg during fertilization. And these enzymes are also useful for the digestion of the stored reserve materials in the germ cells. So the lysosomal enzymes or the lysosomes are also important in the germ cells or in the gametes, that is sperm and egg, fusion of the sperm and egg, that is fertilization. So while entry of the sperm into the egg cell, the lysosomal enzymes or the acrosomal enzymes, that is protease and hyaluronidase, are useful for dissolving egg membrane so as to prepare the entry hole or a penetration path into the egg during the fertilization. So lysosomes are very important in germ cells for the process of fertilization of the egg. So for the penetration of the sperm, the lysosome or the lysosomal enzymes which are present in the acrosome are very important. Then role in a development process. The role of lysosomes in a development processes. Many developmental processes involve the shading or remodeling of the tissues with the removal of whole cells and extracellular material. So during the development, there are several changes during the development of the organisms involve the shading or remodeling of the tissues. Shading, that is removal, or remodeling, that is reformation or the alteration. So many developmental processes involve the shading or remodeling of the tissues in the embryos with the removal of whole cells and extracellular material. For example, the degeneration of the tadpole tail, the degeneration of the frog tadpole tail is produced by the action of cathepsins. So cathepsins, that is a proteolytic enzymes contained in the lysosomes. So the degeneration of the tadpole tail, degeneration or the removal, the degeneration or the removal of the tadpole tail, frog tadpole tail, is due to the action of cathepsins, that is the proteolytic enzymes which are contained in the lysosomes. Then next function, lysosomes involved in diseases and syndromes. Lysosomes are involved in human diseases and syndromes. In certain pathological conditions such as rheumatoid arthritis, silicosis and asbestosis, that is disease produced by the inhalation of silica or the asbestos particles. Silicosis and asbestosis. Silicosis and the asbestosis, these human diseases are caused due to the inhalation of silica and asbestos particles. And gout in which the urate crystals, that is crystals of uric acid, accumulate in the joints. The disease namely gout, in which there is a accumulation of the crystals of uric acid or urate in the joints called as gout. So there is a release of lysosomal enzymes. There is a release of lysosomal enzymes from the macrophages, and acute inflammation of the tissues may lead to an increase in the collagen synthesis. An acute release, serious release, or an acute release of lysosomal enzymes occurs in the states of or in the regions of fibrosis, anoxia, acidosis and shock, and results into increased amount of enzyme in the blood. So this leads to acute release of the lysosomal enzymes occurring in the diseases or in the states of fibrosis, anoxia, acidosis and the shock, and results into increased amount of enzyme in the blood. The lipid soluble vitamins such as A, K, D and E, the lipid soluble vitamins such as A, K, D and E and steroid hormones make the membrane more susceptible to rupture. So one thing, whenever there are certain pathological conditions, occurring in the human being, such as rheumatoid arthritis, silicosis or asbestosis or gout, during which what happens, there is a release of lysosomal enzymes, and this will then lead to the acute inflammation of the tissues and may lead to increase in the collagen synthesis. So this is what the effect of release of lysosomal enzymes in the cell in the pathological conditions. So in a certain pathological conditions or the disease conditions such as rheumatoid arthritis, silicosis and asbestosis and the gout, there is a release of lysosomal enzymes by the macrophages, and this leads to acute inflammation of the tissues, and this may lead to increase in a collagen synthesis. So this is what the role of lysosomes in the human diseases and the syndromes. Then an acute release, that is a large quantity of lysosomal enzymes, when released in the states of fibrosis, anoxia, acidosis and shock, and results in increased amount of enzyme in the blood. So during these conditions, that is fibrosis, anoxia, acidosis and shock, results into increased amount of the lysosomal enzymes in the blood. The lipid soluble vitamins A, K, D and E and steroid hormones cause the membrane more susceptible to rupture, and this can lead to the release of the lysosomal enzymes in the cytoplasm of the cell. On the other hand, the cortisone and hydrocortisone and other drugs that have anti-inflammatory action tend to stabilize the lysosomal membrane. So the hormones like cortisone or hydrocortisone protects or maintains the lysosomal membrane and preventing the release of lysosomal enzymes in the cell. But in a certain disease conditions or the pathological conditions, there may be a release of lysosomal enzyme in the cell and can lead into several human diseases and the syndromes due to the lysosomal enzymes in a variable pathological conditions. Then next function, storage diseases. The lysosomal enzymes also cause the storage diseases caused by mutations that affect lysosomal enzymes. So there are certain diseases. There are certain diseases caused by the mutations which affect the lysosomal enzymes. So several congenital diseases or hereditary diseases. Several congenital diseases. Several congenital diseases or hereditary diseases have been found in which there is an accumulation within the cell. There is an accumulation of substances. There are accumulation of substances in the cells such as glycogen or various glycolipids. So, during several congenital diseases or the hereditary diseases, during due to several congenital or hereditary diseases, there is a accumulation of substances. There is a accumulation of some substances in the cell such as glycogen or various glycolipids, and this will result into the formation of or occurrence of storage diseases, and these are called storage diseases. So whenever there is accumulation of certain substances in the cell leading into the storage of large quantity of these substances such as glycogen or glycolipids called as storage diseases and are produced by the mutation that affects one of the lysosomal enzymes. It means due to the due to the deficiency or absence or lack of certain lysosomal enzymes, there are uh, diseases formed in the cell called as storage disease. So due to the mutation, what happens? Certain lysosomal enzymes are not produced or are not present in the lysosome, and thus the absence of these enzymes will lead to the storage of certain substances like glycogen or glycolipids, and such diseases are called as storage diseases. So these are called as storage diseases and are produced by the mutation that affects one of the lysosomal enzyme involved in the catabolism of the certain substances. For example, in a glycogenosis, for example, in glycogenosis type 2, for example, in a storage disease called as glycogenosis, that is a storage of glycogen in the cell due to the absence of certain enzyme which is responsible for breakdown of glycogen into glucose. So due to the deficiency or shortage or lack of lysosomal enzyme like alpha uh, glucosidase. Alpha glucosidase is an enzyme. Alpha glucosidase is an enzyme. Alpha glycosidase, not glucosidase, an enzyme called as alpha glycosidase. An enzyme called as alpha glycosidase will not be produced in the lysosome due to the mutation, and due to the lack of this enzyme alpha glycosidase, the conversion of glycogen, the conversion or breakdown of glycogen into glucose is stopped, and this results into the storage of glycogen in the cell called as glycogenosis type 2. The liver and muscles, the liver cell or the muscle cells are filled up with the glycogen within the membrane-bound organelles. So, in the storage disease such as glycogenosis type 2, there is a storage of glycogen in the membrane-bound structure in the cell called as glycogenosis. So, this glycogenosis type 2 is caused due to the absence of lysosomal enzyme, that is alpha glycosidase, which will not be produced due to the mutation in the lysosome, and this will result into the storage disease called as glycogenosis type 2. And due to this, there is a storage of or formation of membrane bound organelles enclosing glycogen in the liver and the muscles of the person suffering from storage disease called as glycogenosis type 2. So, in glycogenosis type 2, the liver and muscles cells, the liver cell and muscle cells acquires the membrane bound organelles containing glycogen due to the inhibition or prevention of breakdown of glycogen to glucose due to absence of an enzyme called as alpha glycosidase, and which is not produced due to the mutation in the lysosomes. So, in this disease, that is in a glycogenosis type 2, in a glycogenosis type 2 disease or a storage disease, alpha glycosidase, the enzyme that degrades glycogen to glucose, is absent. There are about 20 diseases. That occur due to lack of certain lysosomal enzymes. There are about 20 diseases that occur due to lack of certain lysosomal enzymes. Then last function, the role of seed germination. The role of lysosome in a seed germination. Lysosomes are found in plant cells. Lysosomes are also found in plant cells. In seedlings, these are involved in the hydrolysis and in the removal of protein and starch during germination. So, during the process of seed germination, lysosomes play important role in the plant cells, that is in seedlings. They are involved in the hydrolysis or the digestion and the removal of the protein and the starch during germination. So, this is what about the functions of the lysosomes. So, we can repeat. I will repeat the lysosomal functions again in a short, in brief account of the lysosomal functions. The hydrolytic enzymes cause digestion of intra and extracellular substances, that is fluid or solid particles brought into the cell by pinocytosis and phagocytosis respectively. So, the important function of the lysosome, that is the hydrolytic enzymes or the lysosomal enzymes, cause the digestion of intracellular and the extracellular substances, that is fluid or solid, that is endogenous material, which are taken into the cell by the process of pinocytosis and phagocytosis, are digested by the lysosomal enzymes. Together with, there are certain enzymes or there are certain lysosomes called as a secondary lysosome, which are formed with the help of mitochondrial material. The part of the mitochondrion can also lead to the formation of lysosome, and that can lead into the digestion of the intracellular substances called as the autophagy. So, autophagy or heterogeneous autophagy or heterophagy. Autophagy or the heterophagy, these are the two type of intracellular digestion taking place in the cell due to the lysosomal enzymes. Then digestion of intracellular structures for energy need, digestion of the intracellular structures for energy need by lysosomal enzymes called as autolysis by the autophagic vacuoles. That is a type of secondary lysosome called as autophagic vacuole, and which is formed by the enzymes or the materials brought from the part of mitochondria to form autophagic vacuoles. So this type of digestion of intracellular structures for the energy requirement or the need due to the lysosomal enzymes is called as autolysis and is caused by autophagic vacuoles or secondary lysosome. Then cellular digestion, when cell dies, the lysosome ruptures. When cell dies, the lysosome ruptures and liberates the hydrolytic enzymes which digest the entire cellular component, and this is a mechanism which removes the dead cells. So the lysosome or the lysosomal enzymes is also responsible for the destruction or removal or the eliminating the dead cells or the worn out cells. Dead or died cells or worn out cells can also be eliminated or destroyed by the lysosomal enzymes called as cellular digestion caused due to the lysosome when cell dies. So when the cell dies, the lysosomes of the dead cell ruptures and liberates all the lysosomal enzymes or the hydrolytic enzymes, and which then digest the entire dead cell or the components of the entire dead cell, and this is a mechanism which helps to remove the dead cells from the tissues. Lysosomes present in the sperms or the male gametes dissolve the egg cell, causing the extracellular digestion, and which helps in the process of fertilization. Lysosomes also help in the initiation of the process of mitosis. Lysosome also helps in a initiation of process of mitosis. So this is what about the functions of the uh, lysosomes. Now we'll explain little bit uh, important points of the topic. That is points to remember. The ER, that is endoplasmic reticulum, is found in almost all animal and plant cells except erythrocytes and the prokaryocytes. Means endoplasmic reticulum is found in all type of eukaryotic cells. Endoplasmic reticulum is found in almost all eukaryotic cells including animals and plants, and ER is absent in RBCs, that is erythrocytes, and prokaryotes like bacteria. There are two basic morphological type of endoplasmic reticulum. There are two basic morphological types of the endoplasmic reticulum, such as rough endoplasmic reticulum, also called as granular endoplasmic reticulum, rough endoplasmic reticulum or granular endoplasmic reticulum, due to presence of ribosomes on the cisternae or the membrane of the endoplasmic reticulum surface, thus appears to be rough due to the presence of ribosomes, and hence is called as rough endoplasmic reticulum or the granular form. The smooth and second type is the smooth endoplasmic reticulum or a granular form. The smooth endoplasmic reticulum having the smooth membrane of the cisternae of the endoplasmic reticulum lacking ribosomes, and thus is called as a smooth endoplasmic reticulum or a granular form. The Golgi complex is supposed to be originated from the endoplasmic reticulum. The Golgi complex is supposed to be originated from the endoplasmic reticulum. The shape of Golgi is quite variable in a somatic animal cells. Even in the same cell, there is a variation with the functional stages. The small vesicles are 400 to 800 angstrom unit in diameter, and they are intimately associated with the cisternae and may show continuity with them. So the Golgi complex is having variable structures or variable forms, that is having flattened sac or the cisternae similar to the endoplasmic reticulum, but are having dilated ends of the flattened sacs or the cisternae. Then there may be small or large vacuoles and the vesicles, which are also accompanied with the Golgi complex with a size of 400 to 800 angstrom unit at the at different stages or at a different functional stages of the Golgi complex. During the maturation of the sperm, the Golgi plays role in the formation of acrosome. During the maturation of the sperm, that is spermiogenesis, transformation of round spermatid to metamorphose or to transform into active motile sperm or the male gamete, called as spermiogenesis. The Golgi complex plays the important role in the formation of acrosome in the head region of the sperm or the male gamete. It is difficult to identify the lysosome under electron microscope because they have no characteristic shape. Means lysosomes do not have a definite shape or size, which is a variable, and thus it is difficult to identify the lysosomes under the electron microscope. The lysosomes are polymorphic. And they show irregularities in a size and the internal structure. The lysosomes are polymorphic, means they are having dynamic structure, and that can be changed time to time on the basis of the functional stage of the lysosome. So lysosomes are polymorphic and they show irregularities in the size and its internal structure. Lysosomes contain variety of enzymes. There are about 40 different enzymes isolated from the lysosome. So lysosomes contain variety of enzymes, and there are about 40 enzymes that have been isolated from the lysosomes. So this is about the functions of the lysosome. So with this, today we'll stop here, and next time we'll continue a new topic. Thank you.